Tuva? Olina?
Yeah.
Sondra?
Yeah.
And that was Tang and Lin. Okay, so you're both here. Hannah?
Yeah.
Mavi?
Yeah.
Jocelyn? No Jocelyn yet. Tuna? There we go. And Chris?
Yeah.
Okay. So what we're going to do today, there's at least one student and one stipendiat who didn't do their self presentations. So and then we're going to get into today's main part, which is what the fieldwork is going to be about. So I guess come on down, you guys, for your presentations. I don't know where yours is. You got it on a pen, memory stick? Okay, we'll let Sven go first.
All right.
And over there. So let me see, computer.
Number 2.
Yeah, in here. Yep, and that was to go. Is there only one available? Yeah. There's only one available, because the other one's closed. So when these things go,
Yeah.
and then when you're done, I think it's only Hassan of the students who hasn't done it. Okay. Okay. Oh, just have a seat. Yes? Oh, Rui's video. Should we wait till he's back? Today? This week? Okay, well, we'll wait till he's back and then I'll do it in front of everybody. Because it's not yet, even though he's got a video, actually the thing is to couple the video videos. Make it like a personal. There's a I'll explain this to you tomorrow when we're doing it. Okay, you ready? Okay. So, yeah, 60 to 90 seconds.
Did it stop?
Hang on a sec. And a sec. Go.
Yes. So hi everybody. I'm Sven. So in short, a few things about me. I like quite a lot of things. I like to plenty of outdoors activities like kayaking, hiking, or snorkeling. I like to do plenty of sports, music, and of course party, like most of the people, I guess. You probably already realized that I'm from France. I'm coming from Parpol, a super small town in Bretagne. And in fact, I'm also half German. But why did it go to the end? Okay. And when I started to study, I went not very far away to Brest. And after 2 years, I thought it was time to leave the country. Went to Edinburgh to finish my master, my bachelor's. And then I started a master in marine biology, first in Bremen, Germany, then Oviedo in Spain, and I did my master thesis in Copenhagen, so I went a bit all around. Then I took holidays and I went hiking and traveling through Europe, and eventually I applied for a PhD in Bergen, and that's where I am since 2 years and a half. So that was a short summary of the last 9 years of my life. What do I do here? I'm working mainly with microbial ecology, so I study microbes and viruses, and I try to understand how they interact between each other and how they interact with their environment. And that's all.
Great, thank you very much. Sven will be one of the stipendiat.
I forgot to say that.
Yeah, I should have said too. He'll be one of the teaching assistants for one of the groups that will be made.
Yeah.
And your turn now. Yeah. Okay, we'll probably have to take a second to load this up. And Hassan, yours is where? Where is your presentation?
It's uploaded already.
It's uploaded already. Okay, let's see if I can get it. Okay, in fact, since I haven't opened the website, we'll take you right after the break. So, okay? Yeah. Is it coming? It's thinking. Oh, you should be on stop. It should be kind of there. Isn't it? Does it come in yet? There we go. Okay.
Yeah. Should I start?
Hang on a sec. Where is it? Yep.
Hi everybody, my name is Mohammad Nizam and I am from Pakistan. I am a PhD student at Civil Exercise Center, University of Bergen. I did my bachelor in Doctor of Veterinary Medicine in 2006 from Pakistan, and then I did my MPhil in the same institute in 2008. Then I did my MS in molecular biology in 2009 from University of Skövde, Sweden. My MPhil thesis was on detection and taxonomy of dust mites and its allergic manifestation. My MS thesis was on effects of replacements and internal deletions of HPPK DHPS enzyme from Plasmodium falciparum, and that I did from Uppsala University. As a research assistant, I joined the National Veterinary Institute in 2010, and my research work was on structural and non-structural proteins of equine rhinovirus and equine arteritis virus. As a PhD candidate in 2012, I joined Rona's group at SEA's Life Research Center, and my PhD project is on oocytes development and maturation in solid glass.
There we go. Perfect. Bang on. Thank you very much. And Tanvir will also be leading one group through the writing of the fieldwork projects. Okay, and now I guess we'll introduce Lars Sørpon. He's from Bergen Kommune. We've been working with him for a couple of years now, and it is the cooperation with Bergen Kommune that has made a huge difference to this course. Huge difference. But the biggest difference that it makes is that it makes your motivation so much more. I mean, basically, we could be teaching you the skills of how to write all this up and make something scientific and publicly available out of this on some lab exercise. But because it is real and because it is part of the municipality, our motivation, your motivation, and theirs changes radically. So when you stop being in the ivory tower and start going out and making connections to the real world, it becomes so much more fun. And it becomes so much more real, and it becomes so much more useful. So after what is it now, 7, 8 years of working with you guys?
9, perhaps.
Yeah, perhaps 9. Yeah. We've got reports that have been on some sites the whole time. So we have a long series of we have a time series, and we have also we have looked at various places, answered many questions, and learned how to work together as 2 completely different institutions. One, the municipal one, which has responsibility to the cities and so on, and the other, the university, which has 2 things basically. The university has 2 main products, teaching and research. And this basically does both at the same time. And this means that you're getting right up to the front line of what you're supposed to be doing, because the answers, the questions they might pose may seem simple, and they're not. And you're going to find out why they're not, but you're also going to find out a whole other series of things. So I'll give the word to Lars, who'll give you a background and so on. And when he gets to the sites, then pay attention to the questions that are going to be asked. Over to you, Lars.
All right, thank you. Good morning. My name is Lars Soholt, and I'm from the Agency for Water and Sewage Works in Bergen municipality. I'm going to talk just a little bit of who we are, what we do, and then I'm going to say a few words about our collaboration with the university and through this course. And then we're going to have a short look at the locations which you will study and investigate this year. Okay, I guess our name gives us away a bit, but these are our three main, or roughly three main areas. We produce water, and we treat wastewater, and we also monitor and make sure the quality of the water in swimming areas is sufficient. So I'll start just shortly with the Bergen water supply and drinking water production. We produce about 36 billion liter of drinking water per year. And almost everyone in Bergen get water from our plants. A few percent, 3–4% have private wells. And of course, our goals are to cover water demand, both for private households and industry. Of course, comply to drinking regulations, water regulations, and have a good quality on the water. And we have 5 large plants in Bergen. Which treat water. We are situated here now. So if you're living in the city center somewhere, you will get your water from Svartedike. This is the larger lake up here, which looks like this. And it's a really nice area for recreation and hiking. That was today's commercial. So, and over to wastewater treatment. We treat about, we have a capacity to treat about 285,000 person equivalents. This is roughly what a given person would produce during 24 hours, included what you could call overhead for industry, etc. Right now we're building we're upgrading 4 new large plants, so actually the treatment capacity is a little bit higher. And of course we also have a lot of pipelines to transport the wastewater to the plants, about 750 kilometers of mains and more than 50 kilometers of tunnels through the mountains around Bergen. So wherever you go, there probably will be some wastewater flowing beneath you. I don't know if it's a compelling thought, but and the discharge from the large, these ones, from the larger treatment plants goes into the different fjord systems. So in 40–50 meters of depth. We also have a few minor wastewater treatment plants. Students in the Bio 300 course have earlier investigated water quality downstream these plants. There are a lot of mini biochemical plants also in Bergen, privately owned, for those who are not connected to the municipality owned wastewater system. Okay, our third area is swimming water quality. Of course, we want the water in the fjords to be as good as possible. So through the summer, from April to August, we monitor all the official swimming sites in Bergen. Once a week we take samples of temperature and bacteria and we put it on the net for everyone to see. And yeah, the purpose is pretty obvious, but of course we want to give everyone information of where it's clean water to swim in, and we also want to monitor for our own sake, so if we find any problems, high levels of bacteria, etc., we know where we have to look for problems and find solution to those problems. And especially in Bergen, we have this rainfall. I don't know if you have noticed, but it's raining a bit. So heavy rainfall often brings up the E. coli count in the water. Also farming activity. There's not a lot of farming in Bergen, but in some areas there is. They give a lot of nutrients to the water. Discharges from sewers and point source pollution. And we use the official European standard for good water quality, which is, I don't know if you can see, but good quality is defined as less than 100 E. coli per 100 milliliter of water. And poor quality is more than 1,000 E. coli per 100 milliliter. And in between it's, well, it's the yellow flag. It's okay to swim, but it's not really good. So I'm just jumping through our collaboration. I will come back to the swimming sites, because one of the groups here will study the water quality at one site. So yeah, we started in 2006. I think we perhaps had the first field work in 2007.
No, I think in fact it was 2006, because I remember coming back from, this is how easy it was to work with the municipality. When I started negotiating here, it was, I knew Håkon Krive, who was with you guys. And I emailed a little bit, and then I came back from sabbatical. I was down in Portugal on sabbatical. I came back on sabbatical on a Monday. I met with Håkon and Anna Konan on the Tuesday. This course started Wednesday. It was that easy to get a plan to work with you, and then we had, of course, write the details and all that stuff. But it was that easy to go, and that was really, really lovely.
Yeah, because we're benefiting a lot from the work you do. It's pretty simple. We have a lot of watershed water courses, I mean small, big lakes, streams, etc., that we want and need to monitor and want to know the water quality. We want to know that our activities are not polluting the water. But we don't have the resources to check every little or large water body. So what we'll be doing, we give the task to you, which we don't have the time or resources to properly go through with. Then you do the research for us, and we read the reports. And now and then we get really good information, so we act on it. So it's very beneficial for us. I will just go, and the course itself is also a success, just academically. Like it says, it integrates, like you said, it integrates research and education and practical work, and also the how the result is presented is a bit unconventional, you can
Now
say.
it's getting
And you will need to use your creative skills more here than any other course.
Yes.
I've taken myself at least. So yeah, you sign up for the right course. And just a few examples how your work is useful for us. For example, swimming areas. Like I said, we monitor the water quality, but we do not have we sample one specific site at one depth, and we don't take the surrounding areas into account always, or we don't have time to take a lot of samples. And the supplement data we get from students is very helpful to see trends in different swimming areas. Also, like I told you about the small wastewater treatment plants, students have investigated how good the water quality is downstream of these plants. And last year,
there was an obvious problem.
yeah, students
Yeah.
found a treatment plant not working properly, so we followed through, and now it's fixed, so it's really good. And of course, water quality in different recipients, the data you collect will give us. Of course, it's just a tiny snapshot of how the situation is when you're out there, but when we get data for a lot of years, the data series will give us a trend, so we get a decent idea of how things are. Okay, and then the last bit of my presentation is your locations for this year. I think these are more or less confirmed.
Yeah.
There will be a slight change, no?
No.
Okay, so that's it then.
What's it, 9 groups, yes?
Yeah.
Yeah.
Or I think it's 10 groups there,
Okay,
9 sites.
right. And we
So, well,
we'll adjust.
yeah.
Yeah.
Okay. So the first one is the aim of the work. You probably figured out now that water quality and ecological status in different water bodies is important for us to find out of. We'll be studying different water quality parameters, the flora, fauna, or the biota bit, and hydrology, and also some of the chemical status in the waters. And some of you, okay, I will just go through the sites because it's easier to see what they will give you of challenges. Okay, this one is the Great Lyngås Lake. It's a saltwater body. It's just 100 meters from here.
Yeah.
If we are situated around here,
Yeah.
so you can go and have a look at it afterwards.
Right.
It's a saltwater body, but it's very limited flux of water in and out during ebb and tide. There's also a river coming into this water body, and as you can see, there's a lot of activity going on around it. There are 2 marinas. There's a lot of construction work, etc. We hope someday it will be a suitable swimming site. So we're monitoring this water body and to find trends in water quality. And I see this is from last year's presentation. I just had to put this in because this is usually we want to have this on the rooster every year. But the Copacabana, I just read that the water is so filthy there sometimes that tourists are advised not to swim there. So
Oh, yeah.
we don't want it to be Copacabana. That's the thing. So. Yeah. Small Lyngås Lake. It's also in the city center. I guess you all passed it from time to time. We're also monitoring water quality there, and finding out whatever lives down there. Could be. There's been introduced some fish, but I'm not sure if they're still alive. There has been some problems with the algal growth there. So, but there are plans to. There are always plans to recover it back to old glory. But we need data to see how it develops. The Steinsvik stream. This is a stream maybe 10 kilometers south of here. There's been a lot of discharge the last few years from construction works. And also there has been, in the stream, there has been some physical blocks. Yeah. So 2 years ago, the 2 guys from the environmental agency, they actually caught fish downstream and then carried it upstream. So there's still a chance for the survival of next year's trouts. And this is a swimming site. It's a bit small there, but you can see during the 90s it was very poor quality. You remember the flags? It's more than 1,000 E. coli per 100 milliliter. And then it was slightly better for a few years, and then it got worse again in 2004, 2008. Then it was pretty good for a few years, but this year it actually turned into poor quality again. And we're not really sure, so why? So we would like to have this investigated. And there are 2 watersheds where we have a couple of waters who are heavily impacted by wastewater, especially from private household septic tanks. This is the water you will be looking at, and this is the whole watershed for the Neston watershed. And it goes also into the Novåsvannet, which I will talk about a bit during the next few slides. Yeah. For those of you not introduced to such a device as a septic tank, this is just a simple drawing. It's simply as you put a tank, dig it into your garden. There's the inlet where the wastewater separates the chunks and bits, the sludge, etc. And then only the water, the nutrient-rich water, goes out. On this slide, of course, the water is infiltrated into the ground, which is a very good solution. But more often, at least in Bergen, it will go straight into the streams and lakes, which of course will cause eutrophiation. And this is also in the same water. We had a project where we have removed some of our sewage outlets. So we hope the situation have improved since last time the students were investigating the area. This is just a map of the area. You can see there are a lot of discharges in this water body. It's a brackish water, and it's pretty shallow, so it doesn't cope well with a lot of nutrients. So some of these have been removed now. So you will get the details for each site when you are being introduced to them, and you will get a contact person with us, which can give you maps and more information on the sites, etc. This one's the same as number 5, or the same kind of task. We have a large watershed also leading out to this brackish water in the Novåsvannet, or Novås Lake. And upstream here, there are smaller lakes which are also heavily impacted by sewage or wastewater from private housings. As you can see, this is it's a bit small. It says it's the EU ecological status. It divides into 5 different categories, and this part is red. That's the worst category. It's really bad ecological quality. This part here is yellow. So first of all, we want to see if it's really that bad, because often these are assessed. They're not really based on in studies. And then when we gather enough data, we know if and where we have to start projects to improve things. And the 2 last ones here, those are the environmental department are in charge of these. I just mentioned them. This is the Göpos watershed. It's the same. It's I mean, it's not really the same as this one, because this is not as impacted by wastewater as the other sites, but there's a lot of work that's been going on. I don't know the details of this water, so I just mentioned this is more general ecological status type of task. Yeah. And lastly, the Høgås River, which is a river north of Bergen, maybe 20 kilometers north of the center. And there's a lot of discharge of wastewater, and also from construction work, road, from the road. And the reason we want to investigate the river is that there's a very rare little creature called Margaritifera. Margaritifera is a river mussel. It's actually nearly extinct in all of Europe. It lives in this river and a few other rivers in Norway. So the environmental agency, HOVA.
Yeah,
Yeah.
we just say HOVA.
Yeah, that's it. There was one person in Bergen municipality who was passionately about these river mussels, and they're actually planning to introduce new ones because they have been hatching small river mussels for 2 years now.
Yes.
Yeah. So it's a very exciting project, but they won't put them into the river unless they're really sure, yeah, it's clean enough for them. Otherwise, they will die.
Actually, it's the oxygen in the sediment.
Oh yeah. It's anoxic sediments a lot of places due to a lot of particles coming into the river. So yeah, we'll have clogging of the pores in the strata.
Right.
Yeah. This is also the environmental department who will be in charge of. So you will get a lot of information if you ask this whole lot.
Yeah.
Yeah. Of course, our main goal is clean water to people and fjords. And I wish you good luck with your project, and we really look forward to cooperate with you with this industry. So, yeah, thanks.
Okay. And just in case, I think there's only 2 sites on that are new. Iglevatn and...
Yeah, let's see.
And these slides will be loaded up to the course website in about 10 minutes or so. Well, possibly a little bit more.
Yeah.
But yeah, Iglevatn is new.
This one, and also this one is new. So, I'm sorry I didn't put a better map here, but number 5 and number 7, they drain to the same large water body, the Novos lake, which is a very interesting lake in itself, since it's also brackish water.
Yeah, stratified lake. Yeah.
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 So, this one, the Høgås Way, this is in the Novos water, so they are connected. In a way. Yeah.
Right. Yeah.
So we really look forward to having the results for these sites.
Now, in case you guys think that it's only a one-way, that we pass things over to them in terms of results, there is a give and take here. He's mentioned E. coli. And how many of you have done microbiology before and sort of plated things out? And how variable are the data? Just tell me somebody. Go ahead.
Very variable. Like, if you mean colony forming units?
Yes, exactly right.
Could say plus minus 50% or
Right.
30.
Yeah. Good point. Okay. And it takes time, correct? And it has to be done in a standardized way. Okay. Which means you guys are not doing it. It is Vannlab, the water lab of Bergen Kommune, that will be processing all the E. coli samples that you collect, so that it is standardized. So we don't have to deal with previous results that are not done in the way that they're supposed to be done. Okay, and that means that the E. coli data that everybody's been working with from the very beginning is in fact generated partly by Vannlab and then by your interpretations. And their interpretations include replicates, because those replications have turned out to be pretty damned interesting.
Yes.
They have. Among other things, one of the because the municipality only has the time and the ability to process one E. coli sample from every water body
Yeah, once a week.
once a week,
Yeah.
right? And the replicate problem will turn out to be, and has in some cases turned out to be extremely interesting, and caused us to think again about a few things. So replication, as in good science, is important here, but also a standardization of methodology. So that is also important for you to be able to interpret your results. We don't want this group did that and this group did that. This is real standardization of methodologies. And then we will also be getting some phosphorus samples. Those are done, you'll be collecting them, but those are also done by Eurofins. It's also a commercial lab. We pay for that. They pay for the E. coli. And then there is the biodiversity. So the outside sourcing of results for phosphorus and outside sourcing of results for E. coli mean that those two data streams are reliable when you're looking back and trying to see why something has happened. So it's not a methodological problem. It might be a replicate problem, it might be something else, but it's not the methodology. That is standardized. The other stuff, everything else you're going to be doing, the abiotic, which means oxygen, temperature, salinity, and the biodiversity, and how you use that, and what else you summarize up, that's up to you. So you will end up doing some samples and things that end up being a dead end, but you're going to be planning it as best you can. And this means that you'll find out afterwards that it's harder than you think. It is much harder than you think. So yeah, I guess that's that.
Yeah.
And I think since we're done about now, does anybody have any questions for Lars while he's here? Not yet. Okay. When we divide, go ahead.
No, I just thought that if there are some translations from Norwegian, at least in the material you will be given, needs to be translated, we will try to at least give a summary of the
Right.
Norwegian text. So,
Yeah. And each group will have at least one Norwegian speaker in it. So that's that. Yeah. So I guess we can take a 15-minute break back here at 10 after, and keep going a bit. Hassan will load up yours. Yep. And is there anybody else who hasn't done a self-presentation? I think that's it. Okay. Good.
right.
Okay. Yeah.
Oh, you need this one.
Yeah.